Anna, do you want to? Perfect, thank you. So. Okay, hello, welcome to the lecture on evolvability. I am Jana. I'm a second-year PhD student at the University of Groningen, and I'll be giving the lectures of this module on evolvability, plasticity, and Evo-Devo. And yeah, let's dive right in. Evolvability. What is this all about? Well, one of the central questions that we ask in evolutionary biology is what determines if an organism can adapt to its environment. And this question is really what evolvability is all about. So evolvability, we can define it as the capability of a biological system to undergo adaptive evolution. In other words, what it is what determines whether or not an organism can adapt to its environment. So why is this something that is interesting? Why should we study that? Well, firstly, evolvability has a lot of practical applications. For example, if we study the evolution of antibiotic resistance, what we're really studying is the capability of a bacterial strain to undergo adaptive evolution. Adaptive for the bacterial strain, not for us. Similarly, if we look at virus evolution, what we're interested in is the capability of a virus to undergo adaptive evolution towards, for instance, vaccine-based immunization. Also, there's a lot of, at the moment, a lot of research into adaptation to anthropogenic change. So this is also another thing where evolvability is really important, because we might wonder what determines whether, for example, an endangered species can adapt to some kind of change to its environment. But beyond these practical applications, evolvability is also really important for understanding evolution, because it gets at the heart of what a lot of questions in evolution are about, namely, what is involved in adaptation? And also one question that is being asked increasingly often is whether we can predict the course of evolution. And if we want to get an answer to that question, it will be crucial to understand what determines evolvability. Okay. So much on the why we should study it. How can we study evolvability? And well, what I'm sort of presenting in this lecture is a way of studying evolvability by studying its determinants, by studying the features that determine evolvability. So the determinants of evolvability, that's defined as organismal features that shape evolvability. And in this lecture, I will be presenting several such determinants. Of course, evolvability is a very, very broad topic. And there are many, many organismal features that affect whether or not an organism or a biological system will be able to evolve. So I won't be able to cover all of them, of course. So forgive me if I'm leaving the one out that is sort of, your favorite determinant or something. But the ones that we'll be covering in this lecture, or at least that we'll mention during this module, is mutation rates, evolutionary capacity, modularity, robustness, and plasticity. But sort of the core of this lecture is really the capacitor, modularity, and robustness. Mutation rates we'll talk about as well, but just briefly and more for completeness sake. And plasticity, I will mention briefly in this lecture, but I won't talk too much about it because we have a whole lecture dedicated solely to plasticity. So if you're interested specifically in the plasticity part, come watch that lecture. Okay, so these are the determinants of evolvability. Let's get started with the first one, with mutation rates. So, and let's start this section by considering an observation, namely that organisms vary a lot in their mutation rates. This is something that has, for example, been observed in cases of experimental evolution. For example, there is this case of experimental evolution with E. coli strains, where they evolved different strains, and 6 out of them evolved hypermutability. So here you can see in different colors the different strains, and on the y-axis, or well, essentially the branch length, reflects the mutation rate. And you can see that some of them have mutation rates that are clearly much, much higher than other ones. Here, the branch lengths are so short that you can barely even see them. So what is causing that difference? Why do some strains end up having really high mutation rates, orders of magnitude higher than other strains? But this is not only something we can see in experimental evolution, it is also something that occurs in nature. Mutation rates vary wildly across different bacterial strains, across different animal species, etc. And mutation rates even vary across the genome. Depending on which part of the genome you look at, the mutation rate that you measure will be different. So why? What is causing differences in mutation rate? Well, there's sort of two answers that are given to that. Firstly, high mutation rates might be an unfortunate byproduct of something else. So for example, it's been suggested that this is something that might arise from a trade-off between replication speed and replication fidelity. If you try to copy your DNA much faster, you're going to make more mistakes. So if you have, for example, strong selection on replication speed, strong selection on dividing the cells very fast and replicating very fast, then you get higher mutation rates just as an unfortunate but unavoidable byproduct. In contrast to that, one can also suggest that high mutation rates are actually adaptive. They are an adaptive response to something. And so some data that sort of supports that is firstly that mutator strains, so strains of higher mutation rates, have been shown to have a higher fitness in various settings, in experimental evolution in the lab, but also natural strains in nature have been shown to have higher fitness.
When I mess with time, it doesn't. What you're telling me is that it already...
And similarly, another piece of sort of evidence in that direction comes from the existence of stress-induced mutagenesis. So some organisms, some bacteria, but also yeast, also some insects, etc., can upregulate their mutation rate when they experience stress. So they do that, for example, by upregulating by switching on a different DNA polymerase that is more error prone, and by switching off those enzymes that correct errors in the DNA. So this is very much an evolved mechanism. Okay, so if high mutation rates are indeed adaptive, are indeed evolved to be high, why? Could it be that high mutation rates are adaptive because they increase evolvability? Okay, well, if we want to answer that question, we maybe first need to consider how mutation rates can evolve. Because mutation rates are a bit of an odd trait in that sense, because mutation rates don't really have their own phenotype that is under selection in the same way in which many other traits do, right? If you have genes that encode the tail length of an organism, those genes get expressed, there is a tail, and that tail is selected on. There might be selection for it to be shorter, for it to be longer, etc. Mutation rates don't so much have their own trait in that sense, but they affect what other traits do. Their trait is how much other traits are changing. So selection on them is very indirect via those traits that they have changed. And therefore, mutation rates evolve in a somewhat more indirect manner. They evolve via something called genetic hitchhiking, where essentially some kind of mutator allele, that is an allele that increases the mutation rate, induces a beneficial mutation somewhere, and then it hitchhikes with the selective sweep of that beneficial mutation. So imagine, for example, that this is your DNA. Somewhere here we have a locus which encodes the mutation rate. Maybe this particular allele is increasing the mutation rate. Then, because we now have a higher mutation rate, we will have a bunch of mutations on this string of DNA. We have some positive mutations, some beneficial mutations, but also some deleterious mutations, some negative mutations. Now, if selection is acting to increase the prevalence of this mutation in the population, it is acting to increase the frequency of this mutation, not only will these mutations spread, but also the mutation rate locus, right? The thing that increased the mutation rate, because they are on the same piece of DNA. They are linked. Similarly, if selection is acting to decrease the frequency of this negative mutation, it will also decrease the frequency of the mutator locus because they are also linked. So that way, mutation rate loci evolve through genetic hitchhiking with those mutations that they have essentially induced. But this also means that the evolution of mutation rates is very sensitive to anything that might break this linkage between the mutation rate locus and, for instance, this positive mutation. So something like, for example, sexual reproduction. If you have sexual reproduction and then you have a crossing over, you have some recombination going on, and these two things get separated, then the positive mutation can spread without the mutation rate locus being dragged along. So therefore, there's actually a lot of debate in the field to what extent mutation rates can undergo adaptive evolution in sexually reproducing populations. And it's not that they can't, but the question is more, to what extent and how exactly does that work? Because it is, of course, more complicated then. Okay, so far so good. The question then becomes whether mutation, do high mutation rates, that should say mutation rates, sorry, my bad. But do high mutation rates that have evolved via genetic hitchhiking indeed increase evolvability or not? And here the evidence is somewhat mixed. You find some papers like this one here, which finds that actually no, higher mutation rates evolve via genetic hitchhiking. They spread together with the adaptive mutations that they have allowed to arise. But this causes the mutation rate to get so high that your mutational load of deleterious mutations also gets really high. And in the end, the population goes extinct. Dead populations are not very good at evolving. So this is not good for evolvability. In contrast, this paper here, for example, found that yes, actually, the evolution of increased mutation rates via this genetic hitchhiking mechanism leads to populations that are evolving much faster and are able to adapt better. And another paper, for instance, found that as the mutation rate increases, evolvability does indeed increase, but only to a certain point, after which, if the mutation rate keeps increasing, evolvability goes back down. So it is it is intermediate mutation rates that confer the highest evolvability. So some papers say low mutation rates give high evolvability. Some say high mutation rates. Some say intermediate mutation rates. The jury, I suppose, is still somewhat out. But the point here is that when we ask whether high mutation rates increase evolvability, this depends on some specific factors. Okay, so the point here is that, of course, high mutation rates increase the incidence of both adaptive and maladaptive mutations. You get more of the good ones and more of the bad ones. And this is linked to this concept that is also called exploration-exploitation trade-off. So if you have a higher mutation rate, you can try out more new variants, right? You can explore many different adaptations, many different phenotypes. But you're very bad at keeping any of those phenotypes. You might find a new phenotype that is really adaptive, but that phenotype is also going to mutate again very soon, in which case you can't actually keep it, so to speak. So you can't exploit the adaptations that you have found if the mutation rate is too high. So this means that whether or not mutation rates increase evolvability or not depends on your current level of adaptation. So consider, for example, this fitness landscape. Where you have the trait value on the x-axis and on the y-axis you have what fitness you get from it. If your population is on an adaptive peak, most mutations will be deleterious, most mutations will be maladaptive. So in this case, increasing your mutation rate would not increase your evolvability because you're just going to fall off that peak and you will not be able to exploit the adaptation that you've already found. In contrast, when the population is currently in a valley, most mutations will actually be adaptive. And what you actually need is not exploiting a trait that you already have because you don't have the good trait, but it is what you need is exploration. You need to be able to explore the space in order to find the fitness peak. So yeah, therefore, whether or not an increase in mutation rate would be adaptive depends on whether you're here, where it wouldn't be adaptive, or down here, where it would be adaptive. Okay, so this sort of brings me to key features of mutation rate evolution that I just want to briefly highlight. Namely, firstly, increases in mutation rate are often transient. Mutation rates might increase when you are in a novel environment where you're not yet adapted, and therefore most, or a greater proportion at least, of mutations would be adaptive. And then mutation rates can be expected to decline again if you're in an environment that you are familiar with, or in an environment that you're already adapted to. And on a similar vein, if you have constantly changing environments, then you have this sort of repeated selection for new variants. In this case, you can actually get constantly high mutation rates because you have repeated hitchhiking events and you have repeated scenarios where indeed the environment has changed again and you're no longer adaptive, and now it would actually be adaptive to increase your mutation rate up to a point. Okay, so much on mutation rates. To sum up, high mutation rates can be a maladaptive byproduct of selection for increased replication speed, or they can be an adaptive result of selection for increased evolvability. We talked about how mutation rates evolve and how this works through genetic hitchhiking with other mutations. And we talked about how the relationship between evolvability and mutation rates is not trivial, because higher mutation rates are not always equal to a higher evolvability. So therefore, the benefit of a high mutation rate depends very much on the current level of adaptation to the environment. Okay, so far on the mutation rates, let's move on to the next determinant that I want to discuss today, namely evolutionary capacitors. Now, to introduce this topic, let's start with a somewhat simplistic view of evolution, where mutation creates variation and selection then depletes that variation. Okay, let's imagine this is our population. We have a lot of different variants present here. And let's imagine we're currently selecting for sort of a yellowish color. So these are the ones that have the highest fitness. They are on the fitness peak. Everything else that is maladapted, that is not well adapted to this, gets filtered out by selection. So these variants will gradually disappear from the population until only the well-adapted variants are left. But if now the environment changes and we select for something else, the population can no longer adapt to that, because those variants have been lost from the population in the meantime. So therefore, it is necessary to wait a very long time until eventually, by chance, we get a new mutation that might be adapted to this new selection pressure. But this is somewhat unfortunate because, the variant was already there previously. It's just that the previous selection pressure removed that variant. So at this point, you might think, wouldn't it be nice if we had some kind of way of storing all of that variation so that it doesn't get depleted by selection? And well, that's exactly what an evolutionary capacitor does. An evolutionary capacitor stores variation. So this is sort of in analogy to electric capacitors, which store electrical charge. Evolutionary capacitors are organismal features which store, so to speak, genetic variation. So they've also been called hide and release mechanisms because they hide variation and then release it. How do evolutionary capacitors work? Okay, so imagine you have a genotype which is translated in some manner into a phenotype, okay? An evolutionary capacitor is some kind of organismal feature which is involved in this translation from genotype to phenotype. And the way it does this is as follows. Let's imagine we have a mutation occurring in the genotype. So we have a new genotype now, which is different. What the capacitor does is that it causes this yellow genotype to still be expressed in the same manner as a green phenotype. So essentially it hides this genotype, it hides it from selection because it isn't expressed. This mutation is not expressed. And therefore it's not removed by selection. So these kind of hidden mutations that are not expressed and therefore not selected against start to accumulate until you have more and more of them. And they are then called cryptic genetic variation, because you do have genetic variation present in this population, even though phenotypically they are all the same. Therefore, it is cryptic variation. So yeah, this is something that is also called canalization, because different genotypes are all canalized into the same phenotype. And this canalization leads to the accumulation of cryptic genetic variation. Then eventually the environment changes, and it might change in some way that disables that capacitor. And now that the capacitor is switched off, all of this variation that was previously hidden, that was previously cryptic, is released. It is expressed. And suddenly we have a phenotypically very diverse population, and selection has a lot of different things that it can act on, a lot of variants that it can act on. Okay. So this is sort of, a schematic drawing of how a evolutionary capacitor works. Let's look at some empirical examples to illustrate this a bit more. And sort of the probably most famous example of an evolutionary capacitor is HSP90, that is heat shock protein 90. Heat shock protein 90 is a chaperone protein, which means it helps other proteins to fold correctly. And this is what allows it to act as an evolutionary capacitor, because even if you have a particular genotype that has mutated to be different, and now the genotype of the protein is different, as long as HSP90 is there to help it fold correctly, you still get the same protein with the same function out of it at the end, even though the genotype has changed. Okay, so with HSP90, what they have shown is that the kind of environmental change that releases variation is a heat shock. If you heat shock organisms during development, that's essentially, yeah, it sort of like overloads HSP90, it overloads the capacitor, and the capacitor can no longer shield all of this variation, it can't hide it anymore, and it's expressed. And they did this, for example, with Drosophila, where if you have this population that you heat, if you have like a strain of Drosophila and you heat shock the larva during development, they start exhibiting all sorts of different phenotypes. Sorry, I think someone has like their microphone still on, and there's like some background noise. Maybe if you could check and switch up your microphone, that would be useful. I don't know, someone is having a lot of background noise. Anyways, I hope you can still hear me well though. Yeah, so the point is that so you get all of these different variants. For example, you have a lot of variation in eye color that is suddenly expressed, a lot of variation in wing length that is suddenly expressed, and so on. But the genotypes underlying this variation, those were already present in this lab population of Drosophila for a long time. It's just that without the heat shock, these variants are not expressed. Okay. Now at this point you might say, okay, yes, fine. Like if you heat shock the population, they start having different wing lengths. What does that mean? Because having like suddenly, like very short wings or something, that's not great. That's a developmental defect. You've basically just affected their development in a way that led to them developing a lot of developmental effects. Why would that increase evolvability? In other words, these capacitors, do they just store deleterious mutations? Or do they also store adaptive variation? Okay, and sorry, I think maybe I'll just throw a quick message in the chat about the background noise.
Okay, that's good.
Okay. You should be able to see my screen again now, although for some reason it has jumped back to the beginning. So I think we were at this point. I hope you can see my screen again in full screen mode and everything. Really sorry for this interruption and for the chaos. I just thought, we should make sure that the audio quality of the recording is actually usable. But yeah, anyways, as I was saying, the question with these capacitors is, because they shield variation from selection, that of course allows also a lot of maladaptive mutations to accumulate. So therefore, the question is, do they really store enough adaptive variation to be relevant for evolution? And actually, it's very neat because they've shown quite recently that HSP90 in particular can indeed facilitate adaptation. And they've shown that with experimental evolution in this insect, Tribolium. So it's a flower beetle. So what they did is that they had experimental evolution with a selection pressure for adaptation to increased light stress. They exposed this Tribolium to increased light stress. And they adapted very fast by evolving smaller eyes, as you can see here on the right-hand side. And the interesting thing, though, is that these smaller eyes, this is not based on a mutation that arose during the experiment. It's based on a mutation that had been present already previously in that strain of Tribolium. It's just that previously that variation had not been expressed. That variation had been cryptic because it was hidden by HSP90. And if HSP90 had not been hiding that variation, probably that particular variant of producing small eyes might very well have been filtered out by selection earlier on, and in which case the population would not have been able to adapt to the increased light stress as fast as they did. Okay, so this is an example where HSP90 stores cryptic variation that is then indeed useful during adaptation. However, capacitors in general do much more than just storing variation. In the next part of the lecture, I want to argue that a sudden release of a range of variation, as you get from evolutionary capacitors, is fundamentally different from the sort of gradual introduction of variation that you get from new mutations. Okay, so therefore, let us talk about capacitors and evolvability. What are the ways in which capacitors affect evolvability? Well, the first point is the one that we've talked about so far, that capacitors store variation and therefore increase the amount of variation that is available for selection to act upon. However, capacitors don't only lead to an increased quantity of variation, they also lead to well-timed variation. So what do I mean with this? Well, if an organism is well adapted and is not experiencing stress, then usually the evolutionary capacitor is switched on and the variation is concealed, which means that when you're well adapted, you're not expressing the variation. However, if the organism is stressed, which is often an indication of maladaptation, that is when the variation gets released, like in the example of HSP90, where it is during heat stress in particular that the capacitor gets disabled and all of this variation is revealed. Similarly, often capacitors are switched off if the organism is in a new environment. So if there's some kind of new environmental input that leads to a release of the variation in many capacitors. So therefore, you get variation exactly when you need it. You get it exactly when it is necessary to adapt to something new. Okay, so capacitors allow you to have more variation, and they allow you to have variation when you need it. Next, capacitors also allow you to cross fitness valleys, to cross valleys in fitness landscapes. So imagine that this is your fitness landscape, and perhaps the population is currently here on this local optimum, and it can't get to the global optimum because there is a valley in between. Without a capacitor, we're pretty much stuck now. But if there is a capacitor, the population might be able to cross the valley whilst it is in the cryptic state, where the trait is not expressed. And then once it has crossed the valley in the cryptic state, it can then proceed up to the global maximum. So this is something that is especially evident when we consider epistatic traits. So imagine, for example, you have a particular situation where if you have the genotype small a, small b, you have, you're at the local fitness peak, so you have some fitness, but not the highest level. You have higher fitness, you have the highest possible level if you have capital A, capital B. However, if you have capital A without capital B, or capital B without capital A, you have much lower fitness. Without a capacitor, it is again very difficult to get from small a, small b to big A, big B, because you can't get either of those mutations on its own. That would be selected against. So essentially, you can only get from small a, small b to big A, big B if both A and B mutate at the same time. And for that to happen is quite unlikely. So it would be extremely difficult to evolve a, capital B. However, if you have a capacitor and the mutations aren't expressed, small a might mutate to big A would be maladaptive, but it's not expressed. So that cost is not experienced and capital A is not selected against. So capital A, whilst shielded by that evolutionary capacitor, can hang around in the population until many, many generations later, B also mutates. And now capital A and capital B are both already present. And then later on, when the variation is revealed and the new mutants are expressed, capital A and capital B are already both present together, and therefore you have been able to cross from small a, small b to big A, big B. So you can therefore see here how having an evolutionary capacitor is very useful, especially when it comes to evolving adaptations that necessitate more than one mutation. Okay. So capacitors can do one more thing, namely they can allow you to have pre-adapted variation through something called incomplete shielding. And in order to explore this, let's look at another classical example of an evolutionary capacitor, this time in yeast. So here in this yeast, we have this protein called sub-35, which comes in two different forms, the normal form and the prion form. And what sub-35 does is that it helps recognize a stop codon. Okay. So when sub-35 is in its normal form, it recognizes the stop codon, we stop at this point, and only the coding sequence is expressed, and we get the normal protein based on the coding sequence. However, sometimes sub-35 folds, like this protein sub-35, folds in a different way, and then it can start to accumulate into this prion form. And when it does that, it can't recognize the stop codon anymore because it's sort of, switched off, which means we don't stop at the stop codon, but we keep going until the next stop codon. And not only the coding sequence is expressed, but also the cryptic sequence that is behind the coding sequence. So we suddenly get all sorts of new proteins because this cryptic sequence is usually not expressed, right? So any sort of mutation might start to accumulate there. And now we get all of these new proteins that all have various different other sequences sort of tacked on at the end. So far, this seems like a pretty standard capacitor, right? You have some kind of mechanism that shields some sequence from some DNA sequence from selection. Here, mutations can accumulate. Every now and then, this variation is released. The new variation is expressed. Now, selection has something to work with. Pretty standard stuff. However, there's something special about this capacitor. Namely, it is leaky. So, what I mean with this is that sub-35 is not really terribly good at its job. It recognizes the stop codon, all right, but it occasionally makes a mistake. Even if it's folded correctly, it doesn't always find the stop codon. So in a very small percentage of cases, something like, I think it was something like 0.5% or something like that, although I'm not sure if I remember that number correctly now, but in a very small number of cases, sub 35, even when it's working properly, doesn't recognize the stop codon, and the cryptic sequence is expressed as well. So every now and then, we get very weak expression of the cryptic sequence, which means that anything that is really strongly deleterious can be filtered out at an early stage. If something is really strongly deleterious, then even weak and occasional expression should be enough to remove that variant. Whereas anything that isn't strongly deleterious should be able to hang around, which means that these variants get essentially pre-screened. We can pre-screen our variation to remove anything that is really bad. These variants are removed, and then when the capacitor is indeed properly switched off and the cryptic variation is revealed, this cryptic variation will be biased towards more adaptive variants, or at least biased away from truly deleterious variants. In other words, this variation, this cryptic variation, is pre-adapted. Okay. So overall, we have seen that capacitors can increase the quantity of variation, but they can also improve the timing. They can allow you to cross fitness values. They can pre-adapt variation. So they do much more than just storing variation. So far, so good. At this point, however, you might say, okay, sure, capacitors are cool. They can do all sorts of things, but are they really common enough to matter? How common are evolutionary capacitors? We've discussed it now in like this one chaperone protein in Drosophila and like some protein that is involved in code and sequence recognition in some yeast strain. But is it really a widespread phenomenon? And well, I think that actually, yes, it is incredibly widespread because capacitance, evolutionary capacitance, is something that is an emergent feature of gene regulatory networks and of plasticity. And what I mean with that is something that we will explore more in the lecture on plasticity. So if you want to hear more on how evolutionary capacitance can result from plasticity, come watch that lecture. For now, I just want to point out essentially that evolutionary capacitors are indeed very widespread because plasticity is widespread and plasticity can act as a capacitor. Okay. This is the end of the section on capacitors. So let me just summarize. Evolutionary capacitors are organismal features that maintain genetic variation by shielding it from selection. And then occasionally that variation can be released, giving selection a lot of material to work with, so to speak. Capacitance can play an important role in evolvability by providing increased amounts of variation, but not only that, it can also, for instance, bias that variation towards a pre-adapted variation or towards well-timed variation, for instance. And finally, capacitance may be widespread in the natural world because it is an emergent feature of gene regulatory networks and phenotypic plasticity. Okay, this was the section on capacitors. Now let's move on to the next one, modularity. What is modularity? Well, modularity is a concept that is used a lot also outside of biology. It's quite a broad concept. It is essentially the ability of a subset of a system to function independent of the context. Sounds somewhat cryptic, but imagine, for example, you are building a toy house out of building blocks. This is a very modular way of building something because you have subsets of that toy house that are individual building blocks that function independently of each other. Okay. So what does this mean in the context of biology, in the sense that is relevant for us? In gene regulatory networks, in networks of genes that regulate each other, that is modular if this network can be divided into modules or sets of genes that are more densely connected with genes within the module than with genes in other modules. So for example, this here is a modular network because you have a bunch of genes that affect each other within the module, but genes in this module do not affect genes in other modules. So they don't interfere with what these genes here are doing. Okay, so therefore this network is modular. Okay, so one sort of topic that is also important for modularity, one concept that is sort of related, is this notion of dissociability or discreteness. You have discrete parts that can be dissociated from each other. Here you have discrete modules in the network that you can separate out quite well. In this toy house, you have discrete building blocks that you can again dissociate from each other by taking them apart. Okay, so how is modularity linked to evolvability? And here I want to point out two ways in which they are linked, two separate ways. The first one is that modularity increases flexibility. And it does so not only in biological contexts. If, for example, you are trying to build a machine, you're trying to, invent a new machine. You don't invent this new machine by reinventing every single part of it. You don't reinvent screws, for example. You don't reinvent the wheel. You don't reinvent the hammer that you need to build it. But instead, you have sort of pre-made parts that you can just sort of put together in a new way in order to build a new machine, right? It would take much, much longer if you had to reinvent these parts each time you wanted to build a new machine. So essentially what modularity allows you to do is that you have a set of modules that can be reused in various ways and combined in various ways, leading to combinatorial diversity. So in the context of evolution, what does that look like? Well, if you evolve a new trait, this is often not done by evolving this trait actually sort of, building it up point mutation by point mutation by point mutation. But you have a few mutations of large phenotypic effect that you use to use a trait that already exists in a new context. Okay, that sounds very cryptic. Let's look at some empirical examples of this. One example of reuse and combinatorial diversity through modularity in biology is the evolution of signaling proteins. So signaling proteins have two domains. They have a regulatory domain, which recognizes when they should be switched on, and you have the catalytic domain, which is what this protein actually does. And these two domains are separate modules that can be reused and combined in different ways. So for example, you might have the regulatory domain GBD, which is GTP binding domain, I think. And this regulatory domain, you might want to have different proteins that are all activated by this, but that do different things. So if you will, for example, want to have a protein that is GBD activated, but polymerizes actin, you don't re-evolve the ability to polymerize actin step mutation by step mutation by step mutation, but instead you have already a catalytic domain for polymerizing actin, and you have a regulatory domain for being activated by by the GBD domain, and then you just have to combine them. And you have a set of different regulatory domains and a set of different catalytic domains that you can combine in various different ways. So this way you can evolve new proteins, you can evolve new phenotypes, not by evolving completely new proteins, but by using already existing proteins that are combined in new ways. So therefore, the modularity of these signaling proteins gives you the flexibility of reusing the parts for new traits. Okay. A second example comes from development, from gene regulatory networks that are involved in development. So for example, Imagine we here have a module that produces a leg. The genes in this module, they are switched on during development, and at the end, when this network has done its job, it produces a leg. So then, in order to produce a leg somewhere in this insect body, this module for making a leg is switched on in a segment. And in order to switch on this module, what we need is antennapedia. That's a transcription factor that switches on the leg module. Now, of course, this is all strongly simplified. Development is a very complicated process, and I'm simplifying it a bit now for the sake of making it more understandable. But yeah, that just is a caveat. And also, antennapedia is actually very good at its job. If you ectopically, that is erroneously, express antennapedia in a different body part, for example, the head, you get legs on the head. So if antennapedia is expressed on the head, then the insect will grow legs instead of antennae. But what having such a module for making a leg means is that if you want to, for example, evolve to have an extra pair of legs on a new body segment in some kind of organism, you don't need to re-evolve the leg there. You don't, build a new leg from scratch through point mutation after point mutation after point mutation. You need only one mutation in order to switch on the leg module in a new place. And this sort of modularity in development has allowed the diversification of a lot of body plans that are essentially just variations upon a common theme. So the arthropod body plans, you can see are all variations of the same theme. So this is an example of how modularity has allowed increased flexibility through evolution of new traits through few mutations of large phenotypic effects. And this sort of flexibility through modularity can then facilitate innovations, such as the innovations of the arthropod body plan. Okay, so this is the first way in which modularity and evolvability are linked. The second way, for more examples of how developmental GRNs and the modularity of developmental GRNs leads to innovation and facilitates evolution, etc., come to the Evo-Devo lecture. We will be discussing this in much more detail there. Okay, the second way in which modularity increases evolvability is by affecting pleiotropy. So, the point of modularity is that most network connections are within a module and not between, right? So therefore, you essentially have modular pleiotropy. Imagine you have, for example, this network. On the top, the network, this one is not modular at all. There is no different modules here, right? And it's very difficult to, for example, change something in this half of the network without affecting the other half on the right, because everything is connected. So therefore, in this case, you have very strong pleiotropy, right? In the lower network, however, this network is modular, which means it's very easy to change something in this left half without changing something in the right half, because it's modular. However, interestingly, these two networks actually have the same number of connections. They have the same number of yellow lines. So what has happened in the lower network is not so much that you have really decreased pleiotropy, you have just rearranged pleiotropy to only occur within the module and not between the modules. So what modularity does is not so much that it decreases pleiotropy, but rather it rearranges pleiotropy in such a way that it becomes modular pleiotropy and is therefore less costly. Okay. Having pleiotropy rearranged into modular pleiotropy allows you to vary traits independently. And this is really where the evolvability benefit comes from. Imagine that the left half of this network codes for your lung, and the right half of the network codes for your eyes. In the top network, it's really hard to improve the lung without changing something in the eye. So you cannot independently perfect both the lungs and the eyes, because improvements in the lung might make your eyes work worse. Whereas in the bottom network, you can change the lung without affecting the eyes. So you can improve your lungs without having the cost of worse eyesight. This also reduces the ruggedness of the fitness landscape. So I think in George's lecture, you talked about how fitness landscapes can be rugged or not. So in this non-modular network, this is what the fitness landscape would look like. Maybe currently you are here on this local peak because maybe your eyes are quite good, but your lung isn't working perfectly yet. And in order to get to the global peak, you would have to improve your lung. However, whenever you improve something about the lung, because of this lack of modularity, your eyes also change and your eyes get worse. So any improvement of the lung makes your eyes worse and takes you down into a fitness valley. So therefore, you cannot get to the global peak where both eyes and lungs are working well. If, on the other hand, you have a modular network, you can improve the lungs without changing your eyes, which means there is no fitness valley and you can just comfortably walk up to that fitness peak. No problems in between. Okay, this means that essentially modularity reduces the cost of change because you can change one thing without making other things worse, and therefore it increases evolvability. Okay, so far we've looked at a few sketches. Let's now look at an empirical example to see whether this might also be happening in nature. And specifically, let's look at some animals that you might already be familiar with, the famous adaptive radiation of Darwin's finches. So one of the key aspects of that radiation is that Darwin's finches differ a lot in their beak shape. And this is what has allowed them to adapt to different food sources and thus to radiate. What is a beak like? Well, a beak has a length. It also has a depth and it has a width. And the interesting thing with how beaks develop is that the beak length and the beak depth/width are regulated by separate signaling cascades. You have, and they are non-overlapping, so they are modular. You have one regulatory network, which is responsible for beak length, and you have a second network, the BMP4 cascade, that is responsible for depth and width. So therefore, because it's modular, you can change length independently from depth and width, because you have two separate modules in beak development. So this means that you can vary the shape of your beak in various different ways. For example, you might have some of the finches that have a short beak and a low beak depth, but you also have other finches that have still a low beak depth, but now with an elongated beak, or they still have a short beak, but now with a high beak depth. So you can see that you can vary the length independently of depth and width. And this allows you to explore a lot of different morphologies, and which in turn allows you to evolve different beak shapes adapted to different food sources, which is the very basis of that adaptive radiation. Now, of course, it's a bit difficult to prove that modularity is indeed what has facilitated this radiation, because we don't have a time machine. We can't go back in time to when this radiation just started and sort of, create a parallel universe where beak development is not modular and then see what would have happened in that hypothetical alternative universe. So therefore, we can't really prove this. But nevertheless, I think this is very strong indication that developmental modularity of the beak facilitated diversification in Darwin's finches. Okay. The next topic I want to talk about is integration, which is very much the other side of the coin of evolvability. But before doing that, let's have a brief break. Okay, see you again in, let's say, 5 minutes. Okay. Okay, so I think you should be able to see my screen again in full screen now. Yeah, welcome back. Last, we talked about how, okay, sorry, the PowerPoint took a second to work again. But yeah, next we will talk about integration. And integration is very much sort of the other side of the coin of modularity. Integration or functional integration, that is defined as the consolidation of multiple functions into a single non-decomposable structural module. So that's a lot of words. But to, sort of give a more informal definition or more informal description, imagine you have two different toy houses, okay? The one on the left, that one is modular. Here you have different modules, different parts that you can take apart, you can decompose this toy house into different parts and then put these parts together in a new structure and build something else with them. In contrast, this toy house is not modular and it is integrated. It is a single integrated item because it is made from a single piece of plastic. You can't take this apart. You can't take it apart and rearrange it in a new way. Nothing like that. It is a single integrated structure. Okay, so I hope that makes it a bit clearer what integration is and how it is different from modularity. Now that leads perhaps to the question of whether integration is the opposite of modularity. And this one is a bit tricky because it kind of is, but it kind of also isn't. Remember this drawing that we had of a modular network? The reason why this is modular is because things within the network are things within a module are densely connected, but things between modules are not densely connected. So the whole network is modular, sure, but within each module, you have integration. Those genes that are within a module of a gene regulatory network are integrated. So you can't really have modularity without integration, because you can't have modularity without modules, and modules are only modules if within themselves they are integrated. So in that sense, integration is not the opposite of modularity because it is almost a prerequisite for modularity. However, on the other hand, if you, for instance, increase the connections between these two modules, and they are now densely connected between the modules, you have integrated the two modules into one module, okay? Because this is now one new module. So if you improve connectedness between modules, you increase integration, but decrease modularity, because now you only have two modules instead of one. This would be less modular than the previous drawing of modularity of the network. So therefore, in a sense, integration is kind of the opposite of modularity, because if you increase integration, modularity decreases. So therefore, it's a bit difficult to really explain whether it's the opposite of modularity or not. So therefore, I prefer to phrase it as integration not being the opposite, but rather the other side of the coin. Okay, so how do modularity, integration, and integration relate to evolvability? Well, when we talked about modularity, we talked about how modularity increases evolvability because it gives you the ability to vary traits independently. So for example, here we have sort of a sketch of one trait versus the other trait, and you can change this in any direction. You can increase both traits, you can decrease both traits, you can increase one trait but decrease the other, or increase the other but decrease one and increase the other. So you can sort of, go in all possible directions here. In contrast, integration also gives you something, but it specifically gives you the ability to co-vary traits. So you can't vary them independently anymore. You can't increase one but decrease the other. However, you specifically have the ability to vary them in the same direction. And this is something that can also be quite important in evolution, because decreased modularity and increased integration can increase evolvability by allowing the coordinated evolution of several traits. Let's give an example for this. When might we need coordinated evolution of several traits? And for this example, let's look at scallops, at a bivalve. What you can see here, feel free to ignore the colors. This figure is showing specifically something about different adaptations, but the colors are not relevant for this point. The point is that we're plotting two traits on the X and Y axis, as we're doing over here, right? And then we're plotting where these traits are with the points. And the two traits in this case are representing the two valves, the left valve and the right valve. And you can see that, of course, these two valves co-vary. If one is further on this axis, so is the other one. We have very much the same shape as we have over here with this oval, right? And firstly, from a sort of, purely mechanistic point of view, it makes a lot of sense that this is how the valves work. Because it's not like you have one set of genes for making the left valve, and then a completely separate, different set of genes for making the right valve. No, that's not how it works. You have one set of genes for making a valve, and then you switch that on twice, once on the left, once on the right. So any changes you make to that set of genes will affect both the left half and the right half because it's the same genes. Of course, again, this is a very simplified way of talking about it, of course. But not only does it make sense from a mechanistic point of view that this is how the kind of distribution that you get, it also makes sense that that would be adaptive. Why? Well, imagine you have a scallop. Imagine you have like a bivalve living on the ocean floor, and now there's selection pressure for this bivalve to, for example, produce a larger shell. For example, there's selection pressure for, just an increase in body size. Now, if this bivalve did not have integration of the two valves, it might have a mutation that affects only, let's say, the right valve. So it grows a shell that is larger on the right half than on the left half, which means the shell might very well not be able to close anymore. That bivalve is probably going to die. So on the other hand, if you have a bivalve that has an integrated development of the left valve and the right valve, any mutation that makes the right valve longer will also make the left valve longer, which means that when this mutation occurs, both halves of the shell become longer and you still get a functioning individual. So therefore, you have coordinated evolution of the left valve and of the right valve, which in this case allows you to evolve and change in your body shape or change in your body size without losing functioning halfway through. Okay. This as just one example of how integration can allow coordinated evolution of several traits. We will have more examples in the lecture on Evo-Devo, so if you find this interesting, come watch that lecture. So to sum up the section on modularity, modularity is the ability of a system to be decomposed into subsets, so it's related to the notion of discreteness. And modular networks in particular can be divided into modules that have high connectedness within modules and low connectedness between modules. And increased modularity can increase evolvability in two different ways. Firstly, it allows increased flexibility through reuse and combinatorial diversity. And secondly, it reduces the costs of pleiotropy by rearranging pleiotropy. And the final point was that we discussed that decreased modularity can also increase evolvability, as integration favors evolvability under certain conditions, when, for instance, we have coordinated evolution of several traits. So overall, this shows that the relationship between modularity and evolvability can be quite complex. This is the end of the session on modularity. Next, let's talk about robustness. Okay, robustness. What is robustness? Well, it's sort of, again, a word that exists also outside of biology. So it's probably a word that you're already quite familiar with and used to. But in the sense of biology, it is defined as the persistence of an organismal trait under perturbations. And specifically important for this lecture is a type of robustness called genetic robustness, that is robustness to mutations. It means that the impact of a mutation on the phenotype is restricted or reduced. So, to use an analogy, this, for example, is a robust house. You can remove some of the wooden planks and the house will still function. Essentially, if the house mutates by removing wooden planks, the organismal trait of being a house is maintained, can persist. In contrast, this is not a robust house. Removing some parts of this house caused it to collapse. So, mutating, removing parts of the house, led to the organismal trait of being a house not persisting. Okay, how does this relate to evolvability? Well, evolvability is the ability to change. Robustness is the ability to withstand change. So you might argue that these are then opposites. Or are they? And at this point, I would like to have a brief 3-minute break where I would like to invite you to think a little bit about whether you can think of any organismal features that combine evolvability and robustness. So let's continue in 3 minutes. Feel free to also discuss ideas in the chat or something in the meantime, if you have any that you want to share with the others. Okay, I think you can see my screen again. Is it in full screen mode? Okay, yeah, so I hope you had some time to ponder this question a little bit, because it is one that is also still very much actively being discussed in the field of evolvability research, and people very much do not agree on whether or not evolvability and robustness are something that can be combined or not. So for instance, some time ago, there was a paper called Evolvability and Robustness, a Paradox Resolved. And then a while later, a different paper came out by someone else called Evolvability and Robustness, a Paradox Restored. So it's still very much actively being debated. But the reason why I asked you to think about whether you can think of features that combine evolvability and robustness is that some of the things we discussed today that increase evolvability actually also increase robustness. So some organismal features favor both evolvability and robustness, such as evolutionary capacitors. Evolutionary capacitors are a robustness on-off switch, essentially. If robustness is switched on, the evolutionary capacitor is switched on and cryptic variation accumulates. The reason why cryptic variation accumulates is because the system is robust, right? The capacitor causes variants, new genotypic variants, not to be expressed. It leads to all of, if you remember the circles that were in different colors, independently of the color of the circle, the phenotype was always green. That is robustness because mutations in the genotype did not lead to changes of the phenotype. So therefore, a evolutionary capacitor increases robustness whilst it is switched on, and therefore cryptic variation accumulates. Then, when the evolutionary capacitor is switched off, so to speak, robustness breaks down, and that is when cryptic variation is revealed. But if there wasn't robustness present in the system during the time in which the evolutionary capacitor is, so to speak, switched on, cryptic variation wouldn't be able to accumulate in the first place. So this is one organismal feature that increases both robustness and evolvability. Another one is modularity. Remember how we talked about how modularity gives you sort of two evolvability benefits. It increases evolvability in two different ways. The first one was flexibility, and the second one was reduced cost of robustness. Reduced cost of pleiotropy. Well, this reduced cost of pleiotropy comes about through robustness. So if we think back to this network, where we said that on if the network is modular, you can change the lung without changing the eyes. This gives you robustness on the level of the whole organism. You can change the lung without changing the eye. In other words, you can have mutations that don't cause the whole system to break down. The system is, in other words, robust. So yeah, therefore modularity also increases robustness as well as evolvability. Okay, these just as two organismal features that increase both. I also want to ask at this point, though, whether robustness in general fundamentally enhances evolvability, not just because there's some features that affect both, but just robustness per se. How does that affect evolvability? And in order to explore this question, we need to look at something called neutral genotype networks. Okay, how do they work? Well, genotype networks, it's a way of sort of mapping the genotype space, of, drawing the genotype space. So how does this work? Well, each circle is a different genotype. The color of the circle is the phenotype that it produces. So here, for example, we have one genotype that makes the color blue, another genotype that makes the color green, another genotype that makes the color pink, and so on. Here we have three genotypes, three different genotypes that all produce the same phenotype, namely blue. Okay? Now the one on the left, in this case, robustness is low. Because, oh, sorry, yeah, one more thing. Each line is a single mutational step. So here, from this circle, one mutation leads you to this genotype, which produces green. Another mutation leads you to this genotype, which produces red. Another mutation leads you to this genotype, which produces yellow. Whereas here, this genotype, one mutation brings you to a different genotype, which still produces the same phenotype though. And another mutation of this genotype also leads to a genotype that still produces the same phenotype, namely blue. Okay, so on the left, this is has low robustness, right? Because every mutation here leads to a change in the phenotype. So this system is very sensitive to mutations, and it is not robust. On the right hand, this is a robust system because half of the mutations here have no effect. So this is robust. Now you might argue that the system on the left has higher evolvability because you can vary things more easily here. Here you have 4 phenotypic variants that are accessible from one individual by single mutation steps, okay? So it is easier to change, it is easy to change something here. Whereas on the right, it's less easy to change something because here you only have 2 new phenotypic variants accessible per individual. So on the left, you might argue this system is more variable. It is easier to vary things here. It is easier to evolve. However, you need to take into account not only phenotypic variants accessible per individual, but you also need to take into account the dynamics of the population. So on the left, let's say we start with a population that is blue. And if we're selecting for blue, then the population is only on this genotype, okay? So the whole population has the same genotype because we're selecting for blue. On the right, if we're selecting for blue, 3 different genotypes can coexist in the population because they all produce the same phenotype. So the population spreads out over these 3 different genotypes. And then we need to extend our drawing because from these genotypes, you might be able to access some more different genotypes. So therefore, on the right, even though per individual circle, you can only access 2 new phenotypic variants, the population as a whole, which is spread out over 3 circles, can actually access 6 new phenotypic variants. So therefore, actually, you have access to more variation in populations that are in networks such as the one on the right. And this thing here, this in the middle, this is called a neutral genotype network. It is a network of genotypes that are selectively neutral because they are phenotypically the same. And they are connected through steps of single mutations. So you can get from one to the other through single mutation. Okay, let's elaborate on this a little bit more by taking into account the action of selection. So you can again see 2 networks here. On the left, we have a small neutral network. Here's only 2 different genotypes that have the same color, only 2 different genotypes that lead to selectively equivalent phenotypes. So this is not very robust. On the right, we have a large neutral network. We have a lot of blue circles that are all connected. So here we have increased robustness. Now, let's say we are, for example, selecting against anything that is sort of greenish or turquoise-ish, so all of this stuff we're selecting against, and we're selecting for red. On the left, you're trapped. You can't do much. You can't get to specifically, you can't get to that red circle because you can't cross all of these green and turquoise-ish points. So it's much more difficult to evolve this adaptation. Whereas on the on the right, you have what is called a neutral pathway towards adaptation. The population is spreading out over this neutral network. Eventually, some variant will have this genotype, and then that genotype is only 1 step away from reaching the highly adaptive phenotype. So therefore, you didn't have to cross any valleys here or anything like that. You had a neutral pathway towards adaptation, which facilitates evolution. So to summarize this argument of the neutral genotype networks, if you have greater robustness to mutations, then more genotypes produce the same phenotype, which means you have a larger neutral genotype network, which in turn means that the population can spread out, so to speak. A larger variety of genotypes can all be present in the population because they all produce the same phenotype. And this means that more different or unique phenotypes can be accessed by the population as a whole, because the population, even though it's phenotypically uniform, is genetically diverse. And therefore, you have greater evolvability. So overall, this chain of reasoning shows that robustness can indeed enhance evolvability. So that brings me to the end of the section on robustness. To summarize, robustness is the persistence of a trait under perturbations, and under genetic robustness in particular, the impact of mutations on the phenotype is restricted. And robustness and evolvability are two concepts that are very interesting to consider together, because on the one hand, you might consider them to be two concepts that are sort of fundamentally at odds because they are opposites in a sense. However, you have some organismal features that favor both evolvability and robustness at the same time, such as evolutionary capacitors and modularity. And secondly, increased robustness itself can also allow the coexistence of different phenotypically equivalent genotypes, which opens so-called neutral pathways towards a larger number of phenotypes, which again facilitates adaptation and increases evolvability. Okay, before we move on to the next section, I also saw a lot of really interesting suggestions for other mechanisms that favor both evolvability and robustness in the chat. I didn't really have time to properly read them all and think about them and discuss them now, but I'll have a look before we end the Zoom session, and if someone is interested, we can bring it up again in the Q&A. But yeah, good suggestions, everyone. Okay, so much on robustness. The final point that I wanted to talk about is phenotypic plasticity. And I only want to talk about this very, very briefly. What is plasticity? Well, it is essentially the expression of different phenotypes by the same genotype in response to some environmental condition. For example, imagine you have this beetle species here. The same genotype can produce beetles with horns or without horns, depending on what you feed them during larval development. Okay, and the question to ask here then is whether plasticity increases or decreases evolvability. And this question is actually very heatedly debated in the field. And if you want to hear more about the various sides of that debate, we will discuss that in the plasticity lecture tomorrow. Okay, that is all I want to say on plasticity now. But to sum things up, evolvability, that is the capability of a biological system to undergo adaptive evolution. And evolvability is shaped by various organismal features, such as mutation rates, evolutionary capacitance, modularity, robustness, and plasticity. Now, the thing is, these organismal features, because they are traits of individuals, they can also evolve. The mutation rate can evolve, plasticity can evolve. These things are all changing. So if mutation rates evolve and plasticity evolve and mutation rates shape evolvability, can evolvability also evolve? Can the ability to undergo evolution itself evolve? And this is a question that I personally find very fascinating. I don't have time to go into it in any more depth during this lecture series, but there's a lot of interesting papers also on that. So if you find that an interesting topic, it is something that I can also recommend some papers on or something if you want to, or perhaps you even want to end up researching this question yourself, who knows. So this is the end of my talk. as I've already mentioned in the previous lecture I gave on sexual selection, throughout I have left references for every concept that I was explaining. I always added like the reference of the paper that it is from. Like, yeah, here, for example, are the papers in which this reasoning is laid out. At the end of my slides, I have a full list of all of the references in alphabetical order if you want to read up on any of the papers I mentioned. I will also upload the slides later. Yeah, these are all the papers that this lecture is based on. And if you have any questions, I very much look forward to seeing them either in Slack or in the Q&A session. Yeah, see you then.